Good morning, students. Today my topic is penetrance, expressivity, complementation test, and pseudo alleles. Penetrance and expressivity are commonly used terms in genetic analysis. Not all alleles present in genes are expressed. So penetrance is defined as percentage of individuals having a particular genotype that express the expected phenotype. If every individual with a particular genotype expresses the trait for that genotype, it is said to have 100% penetrance. An organism is penetrant for the trait when phenotype is consistent with the genotype, or an organism is non-penetrant if it does not produce phenotype generally associated with genotype, or sometimes the genotype does not always produce the expected phenotype, that is known as incomplete penetrance. For example, polydactyly. It refers to persons with extra toes or fingers, which is caused due to a dominant allele. Suppose we examine 100 people having an allele for polydactyly, then it is found that only 65 persons were polydactylous. So, penetrance would be 65%, and this polydactyly has incomplete penetrance. Similarly, the half-black angelfish also have incomplete penetrance. Some of the offsprings never develop half-black coloration. There may be various reasons why individuals will not show associated phenotype although they have a particular genotype. It may be due to epistatic gene or the environmental factors. Moving on to expressivity. It is the degree to which a character is expressed, or we can say that variation in the expression of trait is known as expressivity. In variable expressivity, individuals who carry the alleles for a trait show phenotype but to a varying degree of severity. For example, Waardenburg syndrome has 4 principal features: premature graying, hearing loss, white forelock, and different colored eye. Each family member with this syndrome has same genotype but shows different combinations of symptoms. Waardenburg syndrome show variable expressivity. Some show premature graying while others do not show this character. Similarly, half-black angelfish also show incomplete penetrance and expressivity. These concepts explain why, despite having a genetic code for a specific trait or illness, is not expressed. If there is a gene with low penetrance, it may not be expressed even if trait is dominant, or even it may not be expressed if the trait is recessive and present on both the chromosomes. If an abnormal allele is not shown in a person, but he can pass the allele to the children who may have the abnormality. As we have discussed that expressivity is the degree to which a character is expressed, so expressivity is gradable and hence expressed in terms of percentage. It can range from 0 to 100% and is dependent upon environment, genetic makeup, and age of a person. The major difference between penetrance and expressivity is that penetrance is a quantitative measurement, whereas expressivity is a qualitative measurement. Penetrance is the percentage of individuals of a given genotype exhibiting the phenotype associated with a particular genotype, whereas expressivity is the degree to which the trait is expressed. Penetrance explains how often the gene is expressed, whereas expressivity explains the extent of gene expression. Now this shows the variable penetrance and variable expressivity and both variable penetrance and expressivity. Each oval represents an individual. Now moving on to pseudoalleles. When two genes with similar function located very close on a chromosome and they are genetically linked are called as pseudoalleles. They affect the same character. The concept of pseudoalleles was given by Morgan and Lewis. For example, the red eye color Drosophila has different mutants like white and apricot. They affect the pigmentation, that is, affect the same character, so they are allelic. Moving on to complementation test. It is used to test whether mutations are in same or different genes. Complementation do not occur if mutations are in same gene. It was developed by American scientist Edward Lewis. When two mutations complement each other, they produce wild type of phenotype. Mutations that fail to complement each other are termed as functional alleles. This test for defining alleles on the basis of functionality is called as cis-trans complementation test. If the mutations are allelic, that is, occur at the same locus, then heterozygous offspring will have only mutant alleles and exhibit mutant phenotype. On the other hand, if mutations occur at different loci, that is non-allelic, then the heterozygous offspring will have wild type of phenotype. A heterozygote of two recessive mutations can either have trans or cis arrangement. In transposition, functional alleles produce a mutant phenotype. In cis position, functional alleles produce a wild type of phenotype. Difference in phenotype is called as cis-transposition effect. So, we can conclude that if phenotype is wild type, two mutations have complemented and must be different units of function, or if the phenotype is mutant, the mutation have not complemented and must be in the same units of function.